Good morning, everyone. Luke and I are going to talk to you about a single species, which fits in really quite nicely with what we've heard up until now. This is a marine invasive, arguably one of the worst in the world, called degraded vexillum, the carpet sea squirt, otherwise known as sea vomit or sea snot. Lovely title for it. You can just look at those pictures there and see the impact that it has. These are taken in marinas. So we're going to tell you a little bit about it. So first of all, it's originally native to Japan. It's a colonial tunicate. Distribution now basically worldwide. Hit New Zealand 2001. Netherlands first record in Europe actually in 1991, so it was in Europe before it got to other places. France, Ireland, UK, Spain, Italy, both coasts of South and West United States and Canada, so effectively it's worldwide. The first record in the UK, 2005, north of Dublin. It was then found in Holyhead in Wales in 2008. 2010, discovered in Scotland. Southern coast of England, the bit that we're worried about most because of the proximity to us. Plymouth, Kingswear, Lymington, Cowes, Gosport, northeast of Kent. Basically that's from 2012 to 2015, so very quick to move across the whole of that coastline. It will invade any artificial or natural substrate and structure offshore coastal depths up to 65 metres, so an incredible successful and varied species. It can outcompete, as you can see, forms carpets, so it will smother merle beds, it can take over eelgrass, forms floating mats as well. Yeah, very horrible stuff to find. We were interested in particularly the surveillance because traditional surveillance methods are labour intensive. You have to get out there and you have to look for it, but obviously if you've got a deep water marina, you're not going to know it's there unless you start pulling structures out of the water. Up until last year, we didn't have any records of it in the Channel Islands. We had a kind of a near miss, a vessel that was highly fouled, came into Guernsey in March on a Sunday morning. It had been in Kent, Isle of Sheppey for a number of months. Before that it was a Portsmouth ferry. Both those areas are well contaminated with DVEX, so we were extremely concerned that had we got a contamination threat for this vessel. The bottom picture there shows you all the other species that were also on that boat, so it definitely hadn't been anti-foul for a while, and it definitely hadn't been cleaned before it arrived. When the boat came in, there were certain areas where the oysters had fallen off on the way across, so a lot of these species would have been displaced on transit into Guernsey. So basically we had to set up some surveillance, and I'm going to hand you over to Luke to talk about what we did next.
So I'm going to very briefly go through environmental eDNA, or eDNA. So what is environmental DNA? We're going to basically use eDNA to look for DVEX. Got a nice little diagram on the right here to try and explain what eDNA is. So for this example, I'm going to use fish living in seawater. So got a nice little seawater droplet here. Fish obviously live in the seawater. They're going to leave behind their tissue, skin and scales, free DNA, and metabolic waste. Within those parts, they're going to actually leave their actual DNA, just here. The two main analysis routes for eDNA are either quantitative PCR, and that's where you're looking for one species, or NGS, which is next generation sequencing, and that's where you can look for lots of different species at the same time. So, why is eDNA useful? Arguably cheaper than traditional surveys sometimes. For when you're looking for lots of different species, you might need lots of different experts, but for this you only really need one scientific expert to do the test. Next generation sequencing, you can look for lots of different species at the same time from the same sample. Also with qPCR, if you've got two very similar species or look the same, eDNA can then actually distinguish between the species. As I said, main analysis routes qPCR and sequencing. Lots of applications where eDNA is being used, but one great example is grape nested. Grape crested new populations in ecology.
Okay, thank you. So we had this concern that maybe DVEX had been introduced or actually maybe it was already here. We had no way of knowing, so we worked with Luke and came up with the idea to do some surveillance using eDNA, just to show you where our main harbours are. St Peter Port is the main one, but also St Sampson's and Beauset Marina, private marina, which was blasted out with explosives. Very small marina, but in there you've got houseboats and people are bringing larger vessels from the UK and basically staying there, but also lots of visiting yachts as well. So we wanted to think about where's the main likelihood for DVEX to be introduced. As we had the vessel that we spoke about earlier, we thought we'll start with St Peter Port. The first year we actually spoke to Marines Scotland and they suggested we try settlement tiles, so that's something I'll come on to show you in a second. In the second year we also thought about doing eDNA seawater samples, so basically those two methods were what we started off with. We had a lot of help from CFAS as well because they were able to do the testing for us in the first year. So looking at the settlement tiles, the vessel was in the harbour for a number of weeks, I think up to about 8 weeks. So we decided to start at the entrance of the harbour, but also where the inter-island quay was and on some of the swan pontoons as well. So that was the point where we put out these settlement tiles. Basically they're made of a black plastic with a weight. They're suspended about 1 metre below a structure, and you basically leave them there and you pull them in at the end of the season, and then the scrape samples are sent away. CFAS took samples, but Luke also did the same process testing in the lab. Seawater temperatures are critical for DVEX because this is a species that will release larvae. The data suggests that around about 12, 13, 14 degrees it's likely to start shedding larvae. The other problem is you can also get fragmentation and bits can break off, and then wherever it lands it will colonise. So we were looking at our seawater temperatures, and this year, you know, they were also around about the same sort of period. So the sea temperatures at the moment are about down to 14. During July and August it was up to 17, so well within the range. But we didn't know how DVEX might behave because at that point we hadn't actually found it. So we also asked by CFAS to provide some seawater samples because they were doing a project for DEFRA looking at potentially isolating a whole range of invasive marine species from seawater. So we sent them seawater samples. In 2023 we just focused on seawater, and this was done from the main harbours because we didn't know at that point where we might pick up DVEX. Back to Luke.
So just a little introduction. I work in the State Analytical Lab, which is SAL for short. Our molecular facilities are actually quite new. We actually built them in August 2021 for COVID variant sequencing. And that's basically where we're looking from COVID swabs what variant people have, so Omicron, Delta, etc. The facilities can be separated into 3 distinct labs. We've got DNA extraction here on the right, we've got qPCR and PCR in the bottom right, and then we also have sequencing, which I haven't shown today. You can just about see the little qPCR machines down there, really tiny, don't look very cool. And then on the right we've got a liquid handling robot. Those 3 machines were actually given to us by the pathology department in the hospital, and they were all old COVID PCR equipment. Because everyone's pretty much forgotten about COVID now, we're it's good for me actually. We're looking for new applications, and eDNA is one of them. So a little lesson on DNA extraction. So for the seawaters, the very first thing we're going to do is filter the seawater through a membrane. We're then going to lyse the sample or the membrane to release any DNA from any cells that are left over. And then we're going to bind the DNA to a column using a centrifuge here. Basically where the DNA is going to bind to the column, and then we're going to wash it through with different wash solutions. And then at the end we're just going to elute it. Environmental samples can be very dirty, and they contain high levels of PCR inhibitors which we want to remove. And we also want to purify the DNA from the sample matrices, i.e. the seawater. There's loads of different DNA extraction methods, but we use spin column DNA extraction, which is here using a centrifuge. So, DVEX qPCR. qPCR stands for quantitative polymerase chain reaction. I've got the 2 machines here, little samples within them. To look for DVEX, we're going to basically use forward and reverse primers, and also a fluorescent probe, which is going to bind specifically to a specific part of the Devex genome. So I've got a little diagram down here. So if we imagine these two grey lines are the Devex genome of interest, we've got a forward primer, and then also a reverse primer, which is going to specifically bind to that area. And basically in PCR, all we're going to do is we're going to use the primers and an enzyme called a polymerase to amplify that part of the genome. And as we go through the cycles, basically the polymerase is going to bind to the primer, the probe's going to also bind, and we're going to digest the probe, which is going to release the dye from the quencher, which the machine can then detect. It's a little video, don't quite know how to
Press again.
Again? So this is the liquid handling robot I was talking about. So this is preparing the PCR tubes. So it's going to take the primers, the sample, the polymerase, master mix, probe, put it all together into a tube. It's a nice little video, also quite quick. That's it. I have to say that the machine's actually better than I am. So the machine is going to replace me one day. So the first set of results in 2022, settlement tiles and seawater PCR results. So all the settlement tiles were PCR negative. A portion, like Francis said, were also sent to CFAS. They were also negative by PCR. And then also, St. Peter Port Harbour seawaters were also negative by PCR. So fast forward a year, 23rd of May, expanded surveillance. So we did St. Peter Port Harbour again, but there was more sampling points within the harbour. We also did both at Marina and St. Sampson's, like Francis mentioned. In particular, within St. Peter Port Harbour, we had a positive signal in Victoria Marina, or a suspected positive, because that was the first time we actually found anything. So a week later, Francis resampled. We again got the same result, which you can see here. So the purple line is the PCR plot for the positive control, and then the grey line down here is one of the samples from the Victoria Marina. We also sent the PCR product for Sanger sequencing at Marine Scotland, and they again confirmed that it was Devex, which we were amplifying and found. As Francis says, we didn't do any settlement tiles in 2023.
So alarm bells, as you can imagine, Marine, Victoria Marina is the visitors' marina where all the visiting yachts and boats come in. A few local boats occasionally overwinter there, but it's primarily boats coming from elsewhere, so it looks like this could be an introduction pathway. The next step then was to actually see if we could find evidence. So a physical survey was very quickly arranged, and we were very lucky that at that time Chris Isaacs at the back was doing the same sort of work in Jersey, and we called him over and managed to get him to come and do a survey. I think it was initially a two-day survey, but he spent most of the week because he loves doing that work. Combination of visual inspection, underwater video with a virtual headset as well, so you can see exactly what he's doing, and then physically removing specimens and putting them in bags. Also later on, Chris took them back and did the microscopic analysis as well. He was already aware of what to look for, and he found Devex on 3 pontoons at the top where the purple markers are, very close to the entrance to the marina, but in one specific area. So in a way that was really exciting that Luke had found positive results for the eDNA. Chris came in, he can talk to you about the methodology, which is really interesting, and then he actually found, we subsequently had that tested as well, just to be absolutely sure. So this was in the summer. Here's some photographs to show the sort of work that Chris was doing. As you can imagine, high season, lots of French yachts, people coming and going, so he had to work stepping over mooring ropes, going underneath pontoons with lots of kelp. But you can see the largest specimen, I think, Chris, is that the largest piece there on the right? A sizable chunk, but actually growing around the fan wheel. And he slid that off without any problem at all, which just shows you the nature, how easy it is to displace and disturb any of this stuff. And on the bottom there's some more colonies there. And each of these is an individual colony spreading out. All of it was found on the underside of these pontoons. All of them are heavily fouled. They don't have any anti-fouling on them, and they don't get taken out routinely and cleaned or changed. So that is the big issue for biosecurity.
So again, fast forward a few months, August 2023, resample again. August was chosen because the sea temperature was 17 degrees, which is optimal for DVEX larval dispersal. We again did St. Leopold Harbour, but we added an extra sampling location just outside Victoria Marina. We again did St. Sampson's Marina and also Beauset Marina. So the results, we found DVEX again, or the DNA, in Victoria Marina, which is no surprise. But we didn't find any signs of it spreading outside Victoria Marina, which is good news. And then also from Chris's survey, this tourniquet here, we expected it to be DVEX, and then we tested it through PCR again, and it was in fact DVEX, which is good.
What I didn't mention earlier was that we were worried that it could be dispersing, because I think Chris wasn't able to remove every single colony. He subsequently looked at his video footage and found there was more on the pontoon very close to the exit sill, where the marina empties out every day. He wasn't able to remove that, and we were worried that there would be dispersal and DVEX might be spreading out into the main harbour, which is why we put in the extra seawater testing points. At the moment, we haven't found any signs. It's not a sign of absence, it's just a sign of presence. So at the moment, we're going to have to be doing annual monitoring, but we want to carry on with this eDNA seawater surveillance, but also backed up with physical surveys as well, particularly in that marina. Harbours are finally on board to help us to remove that final piece, and we're hoping that's going to happen next week. Julia, is that the plan? Yeah. So basically, we're looking at what else can we do. I don't think there are any documented cases where marine invasive has ever been eradicated, so we are really looking at containment and biosecurity and education. So this sort of poster is one we found from Marine Scotland. So really we want to be helping our local recreational leisure users and fishermen, shellfish growers, everyone to play their part in preventing possible more reintroductions, but also spread. That's the biggest worry, is spreading within our marinas to wider parts of the island. So this is an interesting one. It's very much all happening at once. Same work is being done in Guernsey as well. I guess it was only a matter of time before it got here and before it was discovered. So far, we don't know of any other islands that have picked it up. Let's hope we can have some success from going back to Hollywood and places where Hollywood. Some big stars on this one. Going back to Holyhead and asking how they've managed to work there, because they had a massive storm incident and all the pontoons were smashed up and it spread all over the coast. But we've got a lot of work that we can do. I think the things to our advantage is we found it early. It's a small, Chris said probably last year, do you think it's only been here, probably the size of the colonies? When the final pit gets taken out, we'll see how much it's grown over the summer. Since it was discovered in June, we'll see what the extent of spread is on that one pontoon. It's a nightmare because it can grow on any surface and it can very quickly get a hold. So yeah, watch this space really. Last couple of slides. Comments? Very quickly go through. So another project we're doing at the moment with Francis is Asian tiger mosquitoes. So Asian tiger mosquitoes, the most likely routes, or because it's not native, onto Guernsey is via the harbours and the distribution depots. So Francis and his team put little traps down, to try and entice them to lay their eggs. So from that we did trap water eDNA, and also if we found any suspicious adults and eggs, we'd also test that by PCR. We just finished that this week and it was all negative, which is good, and also what we wanted. So potential future projects, quite a few. Guernsey quarry water sequencing. And sequencing is where you're looking for lots of different non-native species at the same time, such as fish and reptiles. Signal crayfish sequencing and dwits. Bat roost fecal sequencing for species and prey identification. Also oak and pine processionary moth trap surveillance, and also possibly Asian hornet air eDNA surveillance. A few acknowledgements, which we probably don't have time for, just some references to the method.